Good evening friends, you are most welcome to Think Eyes channel. Today we are going to discuss Practicing Anthropology by MAAN IGNOU, and our topic is Human Genetics and Society. Let us begin the topic. Genetic Screening. Genetic counseling is essentially a communication process that informs prospective parents about the nature of genetic disorder, about the risk of their having a genetically defective child, and about the options available to them in dealing with that risk, or else they can opt to cope with the care of an existing genetically handicapped child. Genetic screening, in contrast, is a routine diagnostic procedure devised to detect those who are carriers of or who are themselves affected by a hereditary disease. Genetic screening applies to populations rather than to individuals. The most widespread application of genetic screening in the United States is for phenylketonuria (PKU). All hospitals in the United States screen newborn babies for PKU by a blood test called Guthrie test. After genetic screening, if both the parents are heterozygous for a genetic disease and the genotypes of the both prospective parents become known, then it is easier to work out the probability of their child, if they decide to have one, inheriting the disease. This can be done through amniocentesis about 2 months after conception. That is, through amniocentesis, the cultured fetal cells are used for determining their karyotype, levels of critical enzymes, and the restriction patterns of DNA. Such an antenatal diagnosis is now available for several genetic diseases and for a variety of chromosomal defects. Such a diagnosis can help the parents to opt for premature termination of abnormal fetus if they so decide. Genetic counseling and antenatal diagnosis provides definite relief to the possible parents at risk and thereby reduce the frequency of genetically defective individuals in the population. However, it is unlikely that these measures would eliminate the deleterious alleles from a population. Eliminate the deleterious alleles from the population. This is so because most genetic defects are recessive and heterozygotes for such alleles. Thus, even after a total ban on reproduction by the homozygous for such recessive alleles, they would remain in the population through the heterozygotes. Therefore, even such an extreme selection would lead to only a slow decline in their frequency. Further, it is not likely that all the couples in any society will willingly submit themselves, at least in the foreseeable future, to these procedures, but genetic counseling has become a routine aspect of medical practice in most developed countries. It has been advocated that defective genes may be corrected through sophisticated genetic techniques either during the early stages of embryo development, embryotherapy, or in specific tissues of the adult patient therapy. Such an approach is referred to as genetic surgery. Embryotherapy involves first, in vitro fertilization of egg, second, production of several copies of the normal allele of the defective gene, third, introduction of this DNA into the zygote or in the cells of the developing embryo, fourth, integration of DNA, preferably in place of defective allele, so that it may function normally. The aim of patient therapy is to introduce the normal gene into the critical tissue of the patient that is affected by genetic disease, that is, the tissue where the concerned gene is required to express itself the most. Example, pancreas in the case of diabetes. The steps involved in the patient therapy are similar to those in embryotherapy, but in this case, cells from the concerned tissue have to be treated in vitro to correct their genetic defects and then reintroduced into the tissue where they may function normally. Techniques for isolation, identification, and multiplication of many human genes are now available, and for many others, they are likely to be developed soon. The techniques for gene transfer in eukaryotes are being refined, and it may not be a great problem in the near future. A suggestion has also been made to use highly specific chemical mutagenes that will correct the defect in the concerned gene. Such a directed mutagenesis, however, is a dream that may be more difficult to fulfill for the patient, and embryotherapies through DNA mediated genetic modification. Genetic screening and counseling may also lead to certain problems. The case the cases of mistaken paternity, the problem of confidentiality, delayed counseling are important among them. Summary: practicing anthropology is one of the significant areas where the information gained by anthropologists is practiced outside academia. Since physical anthropologists have attracted towards human genetics and human genetics has turned out be a central part of the society, the knowledge in some of the areas of human genetics gained by physical anthropologists can be practiced outside academia. Some of the areas consist of serology, hemoglobinopathies, genetic counseling, and genetic screening. It is well known fact that the ABO and RHD blood groups are being used in transfusion medicine. Hence, the history and methodology of blood group polymorphism is presented. The inherited disorder of hemoglobin encompasses hemoglobinopathy and thalassemias. The hemoglobinopathies consist of sickle hemoglobin HbS, hemoglobin C HbC, hemoglobin D HbD, hemoglobin E HbE, hemoglobin O Arab HbO Arab. But widely frequent hemoglobinopathies in India are HbS, HbE, HbD. Genetic counseling is a process that seeks to assist affected individuals and others individuals at risk of getting an inherited condition. It also helps to understand the nature of genetic disorder, its transmission, and the options available for their management and family planning. On the other hand, genetic screening is a regular diagnostic procedure worked out to identify the person who are carriers of or who are themselves affected by a hereditary disease. Genetic screening concerns to population rather than to persons. Thank you very much.